is wherever anyone is watching. As Louis said, I'm Chris Michaels. I'm the team leader at the herpetology section of ZSL London Zoo. And today I'm going to talk a little bit about looking after amphibian larvae and larvae in captivity. Some of this will refer to the talk I gave a couple of weeks ago about water quality. So I won't recapitulate everything I said in that, but I believe the talks will be available via YouTube or already are. So if there's something you want to check, feel free to ask, or you can look back at that presentation. So a lot of amphibians, as their name suggests, are biphasic, and they often have a mostly aquatic larval stage that allows them to adapt to niches completely different to the adult form. And essentially a tadpole has two jobs: to grow as big as it can, as quickly as it can, and to survive until metamorphosis. So they're really just a machine for converting resources into growth. They don't tend to breed other than in metamorphic species. And all this means that amphibian larvae are essentially a different species ecologically from the parent. And that's why I think it's important to have special consideration for them, almost as a completely different entity, as well as the adult species, rather than as an afterthought to adult husbandry. There's a lot of diversity within amphibians about what our amphibian larvae look like between the different orders. So newts and salamanders, anurans, frogs and toads, and caecilians. There are different body plans and different styles of sort of tadpoles, as it were. So whereas salamanders and caecilians are fairly similar to the adult form with external gills attached, frogs and toads have larvae that are pretty much as far away from the adults in body plan as you can imagine. And especially within the anurans, there is a huge amount of morphological diversity and functional diversity. You've got everything from the sort of generalist typical tadpoles in the bottom left through, that's an Elytes militensis tadpole, through megophorid tadpoles with these weird flanged mouth parts in the top left that skim the surface by creating a sort of whirlpool. The central one is an Ascaphus tadpole with its huge mouth parts that allow it to sucker onto to rock faces in fast-flowing streams and feed on algae. Filter feeders like Xenopus, top right, that's a Xenopus longipes tadpole at London Zoo. Or surface feeders like this Agolignus tadpole, bottom right. And that's just a tiny proportion of the diversity of ecotypes within anurans. And this is why it's important to think about the species you're keeping and how to raise it, because they won't all be just like your sort of standard common tadpole, frog tadpole, where you can just chuck some fish flake in and let them get on with it. So I mentioned before there was another presentation about water quality. All the principles that are in that apply to tadpoles, to aquatic tadpoles. And it is particularly important to pay attention to stocking density, just because amphibians can be quite so fecund. But I'll talk a little bit about that in population control later. So I mentioned that this amount of diversity was just the tip of the iceberg. This is a figure from a book about tadpole biology, showing just the array of different guilds of tadpoles, both in terms of the labial morphology, all the different tooth rows and beak forms, and also into the general shape of the tadpole. And all of these different animals have adapted to different environments, and it's important that we know what sort of environment we need to provide to our animals before we breed them. So as well as researching what the adults need, it's important to try and work out as much as possible what the tadpoles need, so that you're not caught short and you have a plan of how to manage the animals if they are to breed. And it's important to bear in mind that some species are well known in the wild. So if you can go out and watch the tadpoles or collect data from the wild, or use published research literature or field reports, etc., to find out more about those animals. But some other larvae are not even particularly well known in the wild. And so talking to other people that have worked with the species or similar species, both those who've done well and those who have failed, is important, so you don't repeat mistakes and you can build on the successes of previous attempts. So what you want to do is replicate the environment as much as possible, and to consider all the different aspects of the physical and biological environment that the animals live in. Are they living in fast-flowing rapids? Are they living in phytotherms? Are they living in stagnant pools? What's the sort of hydrology in the area? What is the underlying geology, and how will that affect the pH and the hardness? How cold or warm is the environment where the animals are living as tadpoles? How much dissolved organic matter and tannins are there? Are we talking about a highly oxygenated mountain stream where the animals are going to be evolved for easy extraction of oxygen? Or are we talking about benthic animals living in the mud at the bottom of a slimy pond? How bright is the environment? Are the animals living underneath rocks where it's completely dark, or are they living in a brightly lit rocky pool, for example? And what's the substrate like? Are the animals living on a variety of substrates? Are they generalists? Are they only found on gravel beds? Are they found on wet rocks? Are they found on plant leaves? All of this can be important to try to copy in captivity. So as well as the physical environment, it's important to think about how you're presenting food to these animals, because getting this right can be really important. And given that amphibian larvae, especially young ones, typically have a very high metabolism, if you don't get this right quickly, you can lose an entire batch of animals before you figure out what was wrong. If it's at all possible to find out what the species feed on in nature, that's really useful. And all of this diversity of mouthparts is basically driven by diversity of diet. So you've got animals that feed on algae, some animals that have adapted to eat carrion, some animals like this ceratophorus tadpole in the top right that eat other amphibians and their larvae, some that feed on live invertebrates like Hymenocyrus, the little Congo frogs. The tadpoles feed on zooplankton. And there's a whole host of other different things. And it's important to remember quite how specialized some tadpoles are. So a good example is Scaphiophorani guttlbii from Madagascar, the tadpole of which is shown bottom right. And this species proved extremely difficult to keep alive in captivity until it was discovered that they actually sort through sand grains in order to find their food. And the only way to keep them alive is to present the food by mixing it into the substrate. Without that, they starve to death because they don't know where to look for the food, no matter how obvious it is in the tank. And Metamorphosis, which we saw top left here with these flanged mouthparts, for example, will only be able to feed on fine particles of food that stick on the surface film of the water. So providing the food there is critical, otherwise it may as well not be provided at all. And there's a whole host of different ways to try out to get tadpoles to eat. And this can be quite a good way, if you aren't 100% certain of how to do it, is to present food in a variety of different ways. So if we create a powdered diet, and I'll look at that shortly, it can be scattered on the surface film. It can be moistened and smeared as paste on rocks and then put under the water so that it acts like an artificial algal film. It can be smeared on rocks in a splash zone, suspended in the water for filter feeders, can be buried in the gravel. Basically, use your imagination about how to get the food to where the tadpoles are looking for it. And you can use the behavior of the animals to give you an idea of what they're doing. A lot of what tadpoles will do is spending their time is looking for food. And if you watch how they're interacting with the environment, that can give you some clues as to where to put the food as well. It's also important to make sure that food is the correct size. So if you're making an artificial diet, making sure that particles are ground to the right size to suit animals that might be filter feeders. Or you might want to culture or collect the right size live food so that very tiny tadpoles actually can fit what they're trying to eat in their mouths. And it's for some larvae, so for example, salamander larvae are often quite aggressive feeders, and they can damage each other. If you just think about things like axolotls, etc., they can take legs off. So think about using either isolation of individuals or forceps to individually feed animals. And there's just a few examples of how to present food here. On the left is surface film for Alites militensis tadpoles that will eat food pretty much wherever you put it. The top right is a defrosted cube of frozen invertebrates for tadpoles that will feed on those. And the bottom right is turning a powder into a paste It can be smeared on a piece of tile, left to dry, and then submerged. And this is quite a good way of working out how much food your animals are eating, because you can do this onto something with a grid pattern on it and calculate the volume of food. It's also important to remember that lots of tadpoles change their diets as they move through development. A lot of species will start being more herbivorous, for example, feeding mainly on algae, and as they grow larger and have to grow structures like full skeleton, limbs, etc., they'll start to require more protein. So watching what the animals are eating can be useful, and providing them with a variety so you can work out what is the best is a good idea. In terms of artificial diets, the sorts of things which are available are a variety of fish flakes, which are a reasonably complete diet, spirulina algae, freeze-dried, dried or wet frozen aquatic invertebrates, specially made amphibian pellets, live aquatic invertebrates for those that require moving prey items, and vegetables. The animal on the bottom right is a Rana arvalis tadpole feeding on some boiled lettuce. It's the age-old way of keeping tadpoles in a jam jar and feeding them on boiled lettuce and spinach, but it can form a useful component of a diet. And you can culture your own as well. For animals that feed on suspended algae or on algae growing in biofilms, this can be grown in high light conditions. So you can see some reactors here for growing suspended algal blooms, which are useful for things like xenoplus. But you can also suspend a light over a tank of water with some tiles or pebbles in it and encourage algae to grow on those, and then cycle them through your tadpole's tank so that they feed on those. For animals requiring live food, collecting from nature can be useful, but this can be also potential for some other things. And collecting the actual diet can be quite useful in that you can get a very wide variety of food for your animals. It reflects exactly what they're adapted to eat, but it has some downsides in that you may import diseases or predators or parasites with that. And it's also important to remember not to go and wreck a completely good natural habitat in order to sustain your animals in the lab. Live food is really important for the vast majority of salamander tadpoles and for larval caecilians, and there are a few species of Anura whose tadpoles feed on live food. And for a lot of these animals, movement is really important, especially for the smaller species, for smaller parts of development where they will not be able to feed based on scent alone and require the movement. And it's a good idea to culture these rather than collect them, because you can then be reliant on a source where you can control. And it's really important to make sure you can create tiny food items. A lot of salamanders, for example, will hatch at just a couple of millimeters in length, and the food items they feed on are almost invisible to the naked eye. And some good groups of organisms to culture are things like Daphnia or water fleas, Artemia, so hatching Artemia salina, you can then use the newly hatched nauplii for newly hatched salamander larvae and grow them all the way up to larger size. It is important to make sure with the Artemia, because they are marine, well, they're salt water at least, to make sure that anything that's not eaten is removed quickly, because it will just die and decompose and kill all of your larvae otherwise. White worms or Enclosures are really useful. They're pictured bottom right. There's two species of those, two different sizes. And these are basically small soil dwelling annelids that can be grown in a cool tub of compost fed on things like bread or instant porridge oats. And they're really, really useful for salamander larvae and caecilian larvae. Panagrellus is a genus of nematodes, sometimes called banana worms or microworms. You can buy starter cultures from these from labs or even on eBay from hobbyists, and they just grow in porridge oats, wet porridge oats or similar, and they measure probably a millimeter or less long, and they're perfect for very small newly hatched larvae. And infusoria is just a general term for small zooplankton, which you can culture by having a piece of potato or similar to that in a jar of water in a bright place, and it will seeded from an aquarium, and you'll get a whole host of tiny little organisms which will feed your very small newt larvae when they've just hatched. You can also purchase some things which are not so easy to culture, things like tuberflex worms and glassworms, which can supplement the live food requirements of diets. For anuran tadpoles, there's a approach of having a sort of generalized omnivorous diet, and this will feed a large proportion of non-specialized anuran tadpoles, or can be modified to feed things like filter feeders, surface feeders, and grazers. This is a recipe which we use at London Zoo. We start with this basic recipe, which is essentially a shotgun type approach of just cramming as many good things into a diet as possible, to make sure that we tickle the nutritional boxes. And this can be tweaked. So, for example, we use this diet with great success for Elates mulatensis, the Mallorca midwife toad, but we applied it for Elates cysternacii, the Iberian midwife toad, a close relative, and found some issues with the protein content, which caused gas buildup in the intestine. And we simply reduced the amount of protein parts, so of animal protein, so reduced the fish flakes, the tuberflex, and the river shrimp, and upped the vegetable aspects of this. And we were able to resolve the problem. So it's also really, really important to consider not just getting amphibian larvae to survive until metamorphosis, but to actually have them thrive until that happens. Because the larger the animals at metamorphosis, the higher the fitness. If you're breeding these animals for release at metamorphosis, especially, their survivorship rates will be massively linked to the size that they hatch at, that they metamorphose at, and that's simply because the bigger you are as a metamorph, the fewer things can eat you and the more stuff you can eat. So it's important that we're not releasing, for example, tiny metamorphs. And if the animals are going to be grown on, the size of metamorphosis is strongly correlated to not only survivorship, but the final adult size and the fecundity, especially of females, of adult animals. So we want to be aiming for high quality animals, not just surviving animals. And one of the things that really mediates this is stress. A lot of amphibians breed in temporary habitats, or habitats where there are threats. And tadpoles basically have to balance the amount of growth they do, so the amount of time they spend in the water and the amount that they eat, against chances of survival. So as things like water quality deteriorate, stocking density increases, or food availability drops, the balance switches, and the HPA stress axis will cause the tadpole to start to metamorphose in order to escape what is clearly a deleterious environment. It's more likely to survive as a rubbish metamorph in its evolution, in the evolutionary sense, than it is to try and hang around in the water and grow when it's not suitable for them. So it's really, really key to make sure that you ensure good water quality with a reasonable stocking density and adequate food provision and suitable amounts of shelter, so that animals have all the conditions they need to stay as a larva for as long as possible and grow nice and large. The stocking density is important not just because of the influence on water quality, but because a lot of amphibian larvae, especially anurans, very little is known about caecilians, and it's possibly the case for chordate larvae, is that they release growth limiting hormones to compete with each other. So the more animals you have in the tank, the more they're going to be chemically competing with each other, suppressing growth and triggering metamorphosis early. So if we imagine we've raised our tadpoles through to metamorphosis, we have to consider how metamorphosis itself affects animals and how to cater to that. It's a really stressful time for tadpoles, especially anurans, because pretty much everything about them changes. Their skeleton becomes calcified, their immune system shuts down and completely reboots to avoid autoimmune problems. At the same time, their major organs reorganize. They go from being often an omnivore or a herbivore to a carnivore. Their entire oral apparatus changes. They grow new adult style keratin in their skin, so their entire skin and water relations change. They lose their gills, grow lungs, and of course they grow limbs in most cases. And generally this is the time when a lot of animals will then leave the water. So this is a vulnerable time for these very small new metamorphosed animals, and it's important that we give them the best chance at getting through it and getting on stabilized on land. So it's absolutely critical that animals that are going to leave the water can do so easily. And this can happen really, really quickly, to go from an animal with larval characteristics to an animal that has lungs rather than gills and can drown very easily, very quickly. So keep an eye on the tadpoles and make sure you act quickly. Shallow water is always a good idea, just to have less of a water column that the animals can drown in. But the means of climbing out the water needs to be species specific. Tree frogs, for example, often climb out emergent vegetation. So making sure that there are overhanging leaves, etc., coming out of the water is important for those. Whereas some other animals, terrestrial things, will tend to crawl up a bank and may require a sloping, sort of a sloping, slanted surface, like the picture in the bottom right. Whereas other animals will be able to climb out onto a floating cork bark. And this can be really important to get right. A good example is golden mantellas and some other mantella species, that if you try to have them metamorphose in a tank with an island in the middle of the enclosure, sort of similar to the one in the picture, especially a floating island, the froglets trying to climb out the water will swim to the edge of the tank and swim against the corner and the sides until they drown. And almost none of them will make it out of the water. Whereas if you provide them with a slope up to a sort of bank side at the side of the aquarium, they will all happily climb out there and be fine. It's really important that enclosures are escape proof. Small metamorphosed amphibians can stick to glass like anything, even climb upside down. So a tightly fitting lid of an appropriate size is really critical to have in place well before the animals get to metamorphosis, just in case you're too late. And humidity and ventilation is really important, especially for some animals like tree frogs, which as soon as they leave the water in nature, go up into a canopy with very high humidity and also very good ventilation. Things can go wrong really, really quickly. So it's important to make sure that the animals have access to the right level of humidity so that they don't desiccate. And again, things like tree frogs can dry to the point where their mucus sticks them to a surface, and then they're unable to get off that surface to go and sit in water, and they will then die. But it's also important to make sure ventilation is good and that humidity is not too high, because skin infections can settle in. And the sorts of conditions that an adult of a species may be able to tolerate for a reasonable time will often kill metamorphs really, really quickly, because their immune system is so poor, their skin is only kind of half changed, and they're in this state where they need absolutely perfect conditions in order to stabilize. So sometimes one can be a victim of one's own success when it comes to breeding amphibians, because many species can produce tens of thousands of eggs. And this is usually too many to successfully rear. Because to do it properly, it requires too much space. And it's sometimes really, really tempting to try to rear everything, because it feels like a waste not to do that, or whatever. So it's important to actually plan for this, because if it goes wrong, you can end up with a load of animals that metamorphose at a tiny size and have low fitness, or worst case, you can overload your systems and end up with mass mortality and fail with everything. And culling is therefore a necessary tool when breeding large numbers of amphibians. The best thing to do for this is to destroy spawn if the species in question has it. But if not, batch euthanasia using, for example, MS222 can be good. Other options are, if the animals are bred for release, to release animals in stages. So a species I work with in the UK in association with another organisation is the pool frog, and that program does a head starting thing where they collect spawn and hatch it, and then as the tadpoles start to grow in the aquaria, when they reach a set density for the size of the tadpole, a few batches are released. So rather than try and get them all through to metamorphosis and outstrip the size of the facility, animals are released in batches to avoid overcrowding. So just a few conclusions. It's really, really important to research your species and to prepare the aquarium or the rearing cups or whatever you're going to use to replicate the natural habitat in as many different ways as possible. To make sure that you are sure that you have space for all the larvae you need to rear to be adequately housed, so you don't end up in a position of trying to raise too many. Make sure the water quality is really good. And again, you can refer to the earlier presentation I've given on that. And make sure that you provide suitable ways for them to come out of the water and to establish on land. And in general, the sort of general idea to think about is to make sure that you treat tadpoles as a species in their own right, and research them and care for them as thoroughly as you would for the adult, and don't treat them just as an afterthought, because you will end up with lower success rates that way. Thank you very much for your time.
Wonderful, thank you, Michael. Chris, I'm sorry. So is the microphone is open, or the chat box for anyone that has any questions for Chris?
I should just note, while anyone might be typing, is that like some of the other presentations I've given, these are just general broad principles, and I didn't want to try and give a recipe for keeping tadpoles or larvae, because no single one appears. So it's best to think about these things and then research the way that people keep certain things. So whether it's dart frogs in plastic cups or whatever, it's best to really look into that particular species. So Juan Chaparro has said, thanks for the talk. Some questions. Can you recommend a brand that sells food for tadpoles and the type of food to cover basic need for tadpoles? It really depends on the species in question. Usually, if the components are, for example, fish flakes or something like that, to just use as reputable a brand as possible and to look at the nutritional content of those things. If you're going to buy some other ingredients, to look at, for example, high quality human products. Yeah, because the country will, as Louis said, the country is important as well. What is worth thinking about is that there are a few products available in the pet trade sold as tadpole food, and I would not necessarily trust those and would rather go with something either specifically prepared for a particular species or to make up your own. So in terms of the fish, sort of the products made for the fish keeping market, which are a good start, things like tetramin are quite good. And that's a sort of a good place to start. But I mean, which species, Juan, do you work with? Okay, cool. So the best thing to do is to try and work out roughly what those things eat in the wild. I think Gastrotheca is quite a generalized tadpole. So something like the food recipe I included earlier in this presentation would be quite good. I think Telmatobius are also quite generalist. So talking to other people that work in a similar kind of area of the world. So for example, in Bolivia, there's a project which is funded by Amphibian, the Bolivian Amphibian, I've completely blanked the name of it. There's works with Telmatobius species, and it'd be worth talking to them, and you can see which products they've managed to source in that kind of corner of the world. I hope that's useful. It's difficult to kind of know what's on the market in Peru.
Also in Peru, you can contact people at the Huachipa Zoo, and
yes,
they have raised Telmatobius culeus too there, and Gastrotheca. A lot of people, different organizations in Ecuador has raised Gastrotheca Rio Grande. so you could talk to them, and if you need, you can provide the contact information of them.
Yeah. So if it was, Juan said maybe a brand from the USA, UK. So for things like fish flakes, Tetra is a good. Tetra products are good, but any high end fish flake is a good place to start with. The other sorts of things are much of a muchness, and they're often sold for as human grade things, so they tend to meet. They tend to sort of to meet the same standards. Olivier, hi Olivier, has said, hi Chris, thanks for this. What can you tell us about water source, tap water, RO, and the water renewal frequency? I guess it depends on the species, but some general thoughts. So I talked about this a bit in the other presentation about water quality, but it really depends again on the species. It's better to try to maintain a chemical environment that is as similar as possible to where the animal lives in nature. Even if it can survive a wider range of things, because a lot of amphibians will adapt to local conditions, particularly as larvae. So if you are planning particularly on releasing animals, it's important to try and match those conditions as much as possible. And it's also important to make sure that the conditions remain as stable and as good as possible. So if it's possible to use biological filtration or a flow-through system, rather than the occasional 100% water change, that's probably a better thing to go for. In terms of the water source, not only do you need to match what the animal has, you need to look at what you have locally, because, for example, tap water will vary massively from place to place and across the year, depending on rainfall, depending on geology, etc. And it will be really suitable for a given species in one area, but completely unsuitable for another. Just for example, from my own experience, in the north of England, where I used to live, I lived in an area with very, very, very soft tap water coming off a peat moorland, and I could keep Mantella tadpoles completely happily in that water straight out of the tap pretty much. Whereas now I live in London, that's not the case. The water is really, really hard, and I have to use other means to keep the species. RO is an interesting one because it's obviously got almost no mineral content. That can be really good for species that live in extremely soft water. So we had a case with Xenopus longipes, where we basically have to keep it in almost pure RO because of the place where it comes from in the wild is almost pure rainwater. Or similarly, things that breed in phytotelms and tree holes, where rainwater, which is very similar in composition to RO, is just accumulating in these places. I hope that's useful. Cool.
All right, Chris, it seems that there is no more question for you right now. Oh, great. There is one more.
Cool. How do you control BD in tadpoles? Okay, so I will preface this by saying I'm not a vet, and probably Luis can talk about this as well. Do you want me to have a stab, Luis, or would you rather?
You go ahead.
So first of all, detecting BD in tadpoles. Very frequently it infects tadpoles, but it doesn't kill them. And the reason, until they metamorphose, and the reason for that is that it infects, the only place it can really infect in a tadpole is the keratinized tooth rows. So it will show up as deformed mouth parts with possibly reduced feeding ability, etc. But you won't necessarily get mass die-offs. And because the fungus really only grows on that part of the tadpole, swabbing for BD, which is the best way to test a population, is important to make sure that the tooth rows themselves are swabbed, which can be really challenging for very small animals, and sometimes you might need to actually sacrifice a subset of the population to get something useful from very small ones. But from larger tadpoles, you can flip them onto their back out of the water on some wet paper towel, and make sure that the swab goes into the mouth between the tooth rows when it's rotated to collect stuff. Then in terms of treatment, there's the usual range of treatments for amphibians for BD are available. So that can be chemical with things like itraconazole or terbinafine, and also temperature. So if the tadpoles will take it, the temperature, the heat treatments which are laid out in the literature. There's different temperatures for different lengths of time, will also kill BD infection.
Yeah, and adding on that, on top of that, itraconazole has been proven to be toxic for some tadpole species. So you always need, in the standard dose, so you always need to try that first on a couple of individuals before you kill the whole batch of tadpoles. So
Yeah.
always try that, and then you can always adjust the dose.
Yeah. I would also note as well that it's important to make sure you are that after the treatment that you retest the animals and then maintain them in quarantine and test them 3 to 6 months again later to make sure that no latent infection has managed to come back. And also with regards to water quality, it is possible to treat animals without sterilizing the enclosure. We've done this with Ambystoma domrillii, which are basically big amphibian larvae because they're pedomorphic. So we basically remove the animals from their tank for each of the itraconazole baths and then put them back in. And what was happening was any zoospores that swimming around in the tank attached to a tadpole, it was then treated, which killed anything, and then put back. And after the requisite treatment length, all of the zoospores that had been in the tank had been sort of sponged up by the tadpoles and then killed. So it was possible to make sure that the water quality remained good while also treating the animals, as opposed to completely sterilizing the tank each way and trading BD infection for deaths due to poor water quality. So Teresa has asked, what is the risk of using tubifex to feed them due to the risk of parasites? So if we're talking about here, yeah, it can be a bit risky, and the reason for that is it is grown in, well, basically it's harvested usually from the outlets of sewage plants, which is great for the worms because it's full of lots of lovely organic stuff for them to eat, but it also means you can have a really high bacterial load, and the same is true for bloodworms. So the typical problems you get with these is not so much macroparasites, but bacteria and sometimes viruses. But things like Vibrio and nasty stuff like that can be in there. The best way to avoid that, if you're going to use tubifex, and it is possible to use it, is to make sure that you get it as fresh as possible, that it's thoroughly washed prior to use. And typically the best way to do this is to get the stuff in, and it usually comes in as a sort of a wet blob on a piece of paper, sort of wet paper, is to soak it in and swill it around in some cold water, pour off all of the mucky brown water and dead worms that come off that, and repeat until the water runs clear, and then leave them overnight in that and repeat the next day. And then after that point, they can usually be fed out, but you should make sure that the water is oxygenated, kept cold, and changed completely every day so that you don't get an accumulation of dead worms and bacteria that are in there. The same is true for bloodworms. It's also worth noting that both of those dietary items can be deficient in some nutrients, so they shouldn't be the entire part of the diet. You can, if you're really worried about it, also treat the live worms with antimicrobial solutions. But there's obviously a risk of that then becoming a toxin to the tadpoles instead of whatever was on the worms. And usually getting fresh worms and washing them, starving them to empty the guts and keeping them clean is a good way around that.
Is there any other question for Chris?
Cool.
All right. Thank you very much, Chris, again
Thanks
for
for the
this
invitation.
wonderful talk. And be in touch. And for the audience, we'll be posting more webinar opportunities. So stay tuned.
Great. Thanks, guys. Bye.
Have a good day and evening for everyone.